Hi everyone, and welcome to the TIA Centre seminar. For those of you that don't know me, my name is Adam Shepherd, and I'm a postdoctoral research fellow here at the TIA Centre at Warwick. I'm delighted to be taking over this, chairing this seminar from Simon. So first of all, I'd like to again say thank you to Simon for his hard work in organising the last set of seminars. I think he's done a brilliant job. And to get started, I'd like to introduce Dr. Mohamed Amgad Tageldin from Northwestern University in Chicago. Mohamed is a physician scientist in training with expertise in computational pathology. He obtained his MBBS degree from Cairo University in 2016 and his PhD degree from Emory University in 2021. His dissertation work utilises crowdsourcing and machine vision to discover prognostic biomarkers in breast cancer using whole slide histology images. Mohamed has received the Digital Pathology Association Travel Award, the Association for Pathology Informatics Trainee Award and Best Poster Award, and Emory's University Graduate Research Award. He is an active member of multiple professional societies in pathology and continues to engage with the community through peer review, education, and outreach. And the title of Mohamed's talk today is Utilising Crowdsourcing Approaches to Create Large-Scale Datasets for Computational Pathology Applications. So Mohamed, if you are ready, please feel free to share your slides and start your presentation. I've just made you a host, so you should be able to now.
Thank you so much for the nice introduction and for the kind invitation. I appreciate it and I look forward. So let me share my screen now. Okay. And maybe I do full screen.
Just to confirm, that you don't mind if we take some questions throughout the presentation, is that okay?
Yeah, that's totally fine.
Brilliant. If there's anything in the chat, I'll ask.
Yeah, sure, no problem. Yeah, I'm expecting a pretty interactive kind of presentation, so no worries at all. Okay, so hi everyone, nice to meet you, and thank you so much again for the kind invitation. So today I'm going to be talking to you about some of the research that I did as part of my PhD under supervision of Dr. Lee Cooper, and the title of my talk is Utilizing Crowdsourcing Approaches for to Curate Large-Scale Datasets for Computational Pathology Applications, and specifically I'm going to be mostly focusing on the work that created the NuCLS dataset. So this was published in a preprint and then later in two separate papers. So the first is that describes the dataset itself and the validation of the data collection approaches and this was published earlier this year in GigaScience. And then the second was sort of a modeling approach for explainable machine learning that I will just touch briefly upon just as a kind of application that may be enabled by the types of data sets that we can create. So first of all, the first question we should always ask, of course, is why should we bother? Like, why do we need to create very large data sets? So let's say we want to work in the space of computational pathology and specifically within the breast. We want to create automatic grading or some form of algorithm that has a bearing on the clinical practice on breast cancer, right? So whenever, when we started this project, our overarching goals were to predict patient survival, see how long people are going to live, maybe apply some of these data sets to predict response to certain chemotherapeutics, and discover biological associations of various types, and discover biological associations of various types, right? But in order to do that, it's actually a pretty complex task, and the reason is, the breast and a lot of solid tumors are pretty diverse in terms of the structures that you see, the visual appearance of the structures that are present in the slide. So first of all, the slides are scanned, so the information is present at multiple resolutions. So there are sort of low resolution information, things like geographic patterns, but then you zoom in and then you see things that are more high power, that you can only see at high power, or at least understand and contextualize when you combine low and high powers. And there are spatial dependencies. So for example, if this is a normal breast acinus, then this gives context to calling this a normal epithelial nucleus, right? So this is normal epithelium, not only because what it looks like at high power, but because of what it integrates into in terms of the context of what's surrounded. There are various things that are even visually difficult for a human being if they're not trained. So that's why we have pathologists. For example, immature fibroblasts. A lot of people would think these look like tumor cells. So there's just a lot of variability in the slides, which complicates the task of really trying to detect these patterns. And why would we try to detect these patterns? Well, any computational algorithm that aims to do computational grading, or that aims to predict survival, discover associations, in order for it to be understood by someone, eventually you will have to get to the bottleneck of linking this to structures that people already know what they are, right? So heat maps can help a little bit with that, but really ideally you want to understand the individual distinct structures, as I will allude to in a minute. One sample famous application that a lot of people like to think is ripe for computational analysis is using tumor infiltrating lymphocytes. So these are basically the immune cells that go and fight the tumor, essentially, right? So if you have the tumor right here, this is the invasive breast cancer, and then you see this really focus right here, and then you see these are tumor infiltrating lymphocytes at the invasive margin of the tumor. Sometimes they go within the tumor itself. So to quantify where they are, what they look like, what markers they have in terms of the CD, like the clusters of differentiation, the membrane markers, all of this has a bearing on how well the patient is going to do. So this is one set of challenges. The other set of challenges has to do with just digital pathology in general. So I don't think I need to elaborate very much in a computational pathology seminar where everyone knows this. So I'll fly by this a little quickly, but essentially the scale of whole slide images is very large. We have things that are composed of 100K by 100K pixels. Typically you can have 500,000 nuclei per slide, especially if it's highly invasive cancer. There's a lot of variability between different centers and different scanners and different places, different hospitals. You have a data problem, which is kind of also unique to computational pathology, in the sense that we have a lot of data that is unlabeled, but we don't have a lot of labels in pathology. So whenever people talk about big data, there is somewhat of people conflating the two with each other, at least in the media, right? Big data implies that we have everything we need to train models, but in computational pathology, we really mostly have unlabeled data, just the slides themselves, which is useful for some weak annotation paradigms that I don't work with. But if you want to do the full supervised machine learning, which uses the data as well as the labels, that is really a big problem in computational pathology in terms of data availability, at least public data availability. And then finally, the fact that this is a critical application. We care a lot about model confidence, we care a lot about interpretability, and these are basically things that you can't compromise with if this is going to have some bearing on patient practice, the clinical practice, right? So the typical computational pathology workflow is you develop a model, right? You collect the data, you generate the ground truth, and then you train some sort of model, and then you validate that this works by seeing if it predicts something useful about the patient or something you already know to be true. What we're focused on is this side of the equation, the data collection and ground truth generation. And we'll touch a little bit about the model training, but mostly we're talking about the data collection and ground truth and specifically how to validate the data collection and ground truth to see which is the right way to do it in a way that is scalable. So going back to the idea of why are we interested in the type of detecting individual elements that I talked about in the first slide. So one famous approach to understanding what the models are doing is to link one slide to one prediction. So to say, for example, this is a slide of invasive breast cancer from patient X. This is what the heat map looks like of where we think the model is looking when it's making the predictions it is. Therefore, we try to infer that this is why the model is making the predictions it is. It's a pretty indirect way of making predictions, and it has its merits. It has a lot of merits, especially that it's the best way to make use of the limited data if you have one label per slide. But it's, of course, in terms of interpretability, it's not the best, because here, for example, it's not very clear why it's focusing, of the cancer areas as well as the stromal areas in between, but not these cancer areas. It's not very clear that the mapping is 100%, and also it's pretty subjective. Someone might say, okay, this may be relevant as well. Why is it not focused on? Why is this more important than this? It's just a little bit more on the subjective end of the spectrum, and suffers a little bit from confirmation bias and selection bias. So while it's something that is very important and has its merits for a range of applications, we thought we would apply something that is fairly old style, but applied in this context is new, which is the idea of concept bottlenecking. So I say this is old style because the classical computational pathology workflow really was mostly about this. People would detect nuclei and detect individual chunks of cancer nests, right? And then they would calculate their individual properties, like what is their shape and texture and geospatial relationships. And then they would use this to predict various things about the patient. What was done in the past, though, before the deep learning revolution, I think in 2012, if I'm not mistaken, is that, and even now in some limited applications, what it was done is mostly by manually. So you would say, I would say if a nucleus is large enough, or if it looks ugly enough, or if it's in the right place, then therefore this is a cancer nucleus. And as you can tell, because it is very manual, first of all, it's not very robust, because what you think is important may not be translated well across different centers. But also, it's quite subjective. It's hard to know what really matters, and it's hard to know that to translate what pathologists subjectively consider when they're making these calls, when they're saying this is cancer because it has prominent nucleoli and it looks ugly and it has a feel that it's a cancer nucleus, or that there's variability in the tubule formation. A lot of these things, it's not immediately apparent that you can translate them to things you can detect yourself and be sure that this is the best way to measure it. So for example, if you say, well, I'm going to look at the largest nucleus, well, this is fairly robust. But if you say the nucleus with the most rough texture, well, it's not very clear that roughness is what we mean by heterotopic chromatin. So what we're doing is we're applying the same concept, but using deep learning. So we're detecting these intermediate properties or intermediate patterns, and then using those to detect the final task that we care about. So this was from the concept bottleneck paper that sort of formulated this most famously is, let's say you want to look at whether this is some bird species, then you would use the convolutional neural network to detect the wing color, the tail color, beak length. So you come up with various features of interest, not as something intermediate to, like, not saying the wing is the elongated thing that is towards the posterior end. No, you let the deep learning model, you let the computer itself learn how to detect these things, but it detects these things intermediately, and that's that's the concept that you're later going to put in a simple classifier, just a linear model or something like that, to detect the final pattern. So the advantage here is that it's combines two things, right? It's the best of both worlds. You have the convolutional neural network doing the heavy lifting, and then you have a simple classifier doing the final prediction. Okay, so this is just to say that all of this was done in the backdrop of these collaborators, and was done using the Digital Slide Archive, which is a virtual slide annotation platform that we developed in collaboration with the company Kitware. Yeah, so it's basically a platform where people can log in from anywhere around the world. They can place their annotations from a set of predetermined classes, and then it immediately just gets saved to the server to create the data that we need to train these CNNs and to validate that they're detecting the correct concepts. Okay, so end result is we created essentially a tissue atlas, if you will, that is composed of both sort of the low power patterns, the region annotations, and that was in 2019, and as well as the nuclei, the individual nuclei. And the way we did this is essentially we validated a framework where we organize medical students and junior pathologists, and junior pathologists are defined as pathology residents who haven't completed 2 years of training. So if they are third-year residents, they clump with the senior, quote unquote, or trained pathologist. In any case, so we've got non-pathologists with minimal experience, and then we've got junior pathologists and senior pathologists with more experience. And in order to create these annotations, drawing these boundaries so that we can train the computer and validate that it's doing the right job, that's a very manually intensive process. That's a laborious process. And we can't really be asking pathologists to do something like that, this extremely repetitive task, and expect that this will be repeatable. We may do it once, we may do it twice, but it won't be a robust process that we can repeat over and over again for different projects. So something has to give, and we thought maybe we make use of crowdsourcing. We have a pool of people who have exposure to pathology through their med school. That pool is not in any specific location, but they're worldwide. We can tap their interest in advancing their careers and doing research and getting their names on papers and their names on data sets and so on. And then make sure that they're doing a good job by having supervision of pathologists. So we're not the inventors of crowdsourcing. This is a very old or relatively old technique and procedure, but really our contribution was mostly in validating doing the crowdsourcing in a setting where the data itself is quite specialized, and doing this in a structured fashion where we incorporate the feedback from the pathologist, and we also validate a lot of this. So in terms of breast cancer and in terms of H&E or hematoxin eosin stain slides, this was the first work to kind of apply crowdsourcing and adapt it to the specific problem that we're working at. So these were two different projects. The first one I will not really talk about today for time constraints, which is basically we ask people to annotate regions, geographic patterns. The second one is what I'm focusing mostly on, which is nuclei. So the way this works is you ask the junior pathologist, here depicted in pink, to annotate things on this shared annotation platform. And then they would, everyone is on a workspace. In our case, we use Slack. They would ask questions, and then the pathologist would give them feedback and say, well, this is X, this is Y, and here's why I think it's a plasma cell or it's a tumor or so on. And then we got a couple of coordinators who would sort of incorporate the feedback and then work with the pathologist to triage the slides by which ones need correction, which ones need improvement, based on what the pathologist gives, the feedback they give on the slides, where most of the manual work was done by the junior participants. Okay, so I'm going to just skip this. This is irrelevant. So let's let's take a look at the specific project that we worked on. So we have we decided to focus on a bunch of classes. We said, all right, let's do a bottom-up approach. Let's give them every single potential class that they can pick from, and just let them choose. So they can pick tumor, fibroblast, lymphocyte. So this is the cancer itself. These are things that generate the matrix around the cancer. These are cancer fighting cells, and then various other things that may be a little less prevalent in the slide, and therefore maybe a little more difficult for the junior members to really reliably detect. And then we thought maybe like other machine vision data sets, we impose some sort of hierarchy, at least when we do the analysis, where we can think of superclasses. So in case of cars, you can think of individual cars, Corolla, Camry, Verna, whatever, and then eventually they're all under the category of cars, right? So this is kind of similar. We impose some sort of hierarchy so that maybe we thought this will will improve the variability between how well people detect various things. So the reason I told you about the region annotation project is we didn't want to have the participants draw the individual boundaries around the nuclei. Now, nowadays, there's something called NuCLS that I'm sure you guys are familiar with. You developed it. It wasn't there at the moment we did the work, and we also had in our individual approach to this. And what we thought is we wanted to have the participants sort of click on things that already have a segmentation boundary. So essentially, we said, if we can initialize them with nuclear boundaries and classifications that may be wrong, but if they are correct 10% of the time, then we save them the effort involved in detecting these and drawing these boundaries all of the time, right? So essentially, we would give them, they would have access to the previous data set from the sort of bird's eye view of the region suggestions, and then they would also have access to just very weak suggestions of what the individual nuclear boundaries are and what their classifications might be. Now, we might say this biases the participants in one way or the other, and we actually test for that later, and we show that it's actually not that big of a bias, and it actually improves their classifications more than it biases them in any wrong way. So anyways, this is what the participants were asked to do. So this is the background on which they are working, and then if they see something that they like, they just click on it and place a dot. And then the color of the dot is what they think the class is. So it's a very quick process. They change let's say they are now annotating all the lymphocytes in this field. They will change the classification default mode, and then they just click all the lymphocytes. And if it's already, so again, this suggestion, it doesn't carry over. This is just for them to see in the background. They still have to place their dots just to confirm or not confirm that they agree with this. And then let's say the boundary is pretty messed up. Like here, these are two different nuclei that the segmentation, the initializing boundary didn't really separate. So what they end up doing is that they place bounding boxes. So again, we want to save time. So you'll say this is a little bit peculiar, right? Is we have a mixture. The end result is wherever they clicked, we would have the segmentation boundaries of the nuclei. Wherever they place the box, which is very easy to do, they just drag and drop, right? Drag. Then we would have a box. And our rationale was we wanted to prioritize scale. And we also thought that it's quite easy to modify what was the best model at the time, mask RCNN, to make sure that it actually takes in this mixed form of data set if we wanted to, or if people wanted to just convert the segmentation boundaries to bounding boxes, they could. But we really wanted to prioritize scale over really asking people to spend all the time to draw things manually, because it really does take a lot of time.
I just asked a quick question there, sorry.
Yeah, of
So,
course.
so in terms of the initial sort of guesses for like the nuclear segmentations, so where exactly do those come from then, if they're not coming from nucleic?
Exactly. That's a great question. So this is how we are getting those suggestions.
Okay, brilliant.
So the way we get the suggestion is, let's start here. So we have an image, an RGB image. This is just a field of view, and then we apply some heuristic image processing based model. So classical machine vision pre-deep learning, right? Just classical machine vision based on Gaussian modeling and things of that nature. So this will look at areas of high contrast, and I'm picking here a particularly difficult field with lots of clumped nuclei just to illustrate the point. But usually it's better than that. Usually it's much better than that. But to illustrate the point, we initialize this with traditional machine vision approaches. And then what we do is, now that we have this big data set of quite low to intermediate quality nuclear segmentations, and they're only segmentation boundaries really, is we train, we import the low power region annotations from the previous data set, from the previous crowdsourcing project, where people would look at like this zoomed out view and would say, all of this looks like cancer, all of this looks like stroma, these are collections of lymphocytes. Now, the low power view is not always going to be accurate in the high power. For example, we can have a stromal area with some lymphocytes. You can have some cancer cells that are sort of wandering around, and lymphocytes are scattered a lot in stromal areas, not only in areas which the participants in the previous low power view considered like a collection, or just a sea of lymphocyte, right? You can have individual ones that are floating on their own. But we thought this was a pretty good way to initialize the classification of the nuclei. So now we have the segmentations, and we have the classifications just by region membership. And then what we end up with, we also impose very simple compatibility types of constraints, but I won't be spending too much time on that. So what you end up with is this sort of low to intermediate accuracy type of initializing data set. Now, what we consider is, can we actually improve on this? And then the answer is yes. What we thought we would do is use mask RCNN as a function approximator. And the idea here is, sometimes you won't have like here, the segmentation algorithm failed to place boundaries, nuclear segmentation boundaries in this area. It's just the contrast was too high in the image, or the too high in this area that it's sort of drowned out this area. Whatever the reason, it failed in this particular area. But we thought, all right, can we use the fact that when you fit a deep learning model to the training data set, it is not memorizing the data. Even if it could, it wouldn't. There's a limited capacity of that model. What it is really, it is fitting a line, or fitting a nonlinear approximation of the underlying representation of the data, right? Just like if you have like a bunch of noisy points, you can fit a line to it. So we used mask RCNN, which is a nucleus segmentation and classification algorithm that was state of the art, to essentially find something that fits the data, the training data. And what we end up with is something that is better than the input, right? It's better than the input in the sense that if there's something missing, it would be filled out, because again, you train on this and then you apply it to the same data it was trained on. It will eventually have to, it smoothes out the noise. So this was what the participants saw. Now, what they saw is a smoothed out representation of the data set that was created by combining segmentation boundaries from heuristic image analysis with manual segmentation of region boundaries. And then they would use that and do the annotation that I talked about earlier. And the way they annotate is they choose a class, like I mentioned earlier. And then once they choose a class, they just either click or make a box, click, make box, click, make box. So it's pretty quick in terms of annotation. They don't have to spend time drawing boundaries. Okay, so what we said is, all right, so how do we structure this so that we actually validate the approach and how good it is and so on, is we said, oh, sorry, one more thing I want to mention is the fact that we use heuristic nucleus segmentation using image analysis is only specific to what we wanted to do. Is we wanted something in invasive breast cancer, and there weren't a lot of data sets that had the exact purpose we wanted. But if we did have data sets that, or if this was something we wanted to prove, we wanted to see how we do in the difficult scenario where we initialize with machine vision, just classical machine vision. But you could also see that we could have initialized this with deep learning approaches from other data sets, or transfer learning from other cancers, or things of that nature. If I do extensions of this project and other applications, that's what I would actually do. But here we just want to see the more difficult case. So we thought we would have two levels of data sets. One is what I just described, where there's feedback. This would generate the majority of the data that would be useful to actually train models down the line, because this is one person gets one field, so you can get scale, right? And the field eventually gets corrected by a pathologist, or at least they give a quality score. But in order to validate the approach itself, we needed to also create multi-rater data sets, limited in scope in terms of the number of fields of view. By the way, FOV here is a field of view, like a small area. And that would be where we have everyone annotate the same thing, or at least most people annotating the same thing. So we would have the pathologist and the non-pathologist annotate the same area, and then we can infer what the aggregate pathologist classification is, what the aggregate non-pathologist labels are. So we call it the p-truth, as in the pathologist truth, versus the NP labels, or the non-pathologist labels, because in this case, we consider this to be the truth and this to be a label of some sort, because this is the aggregate of people who are not experts. So even if there is wisdom of the crowd, it's not going to capture, for example, classes that are not common. It's not. There are things that even wisdom of an uneducated crowd or non-expert crowd can't compensate for. And then we created 3 levels of control. So a lot of this paper was talking about controls, really. We had the evaluation data set, which is essentially using the exact same workflow that I talked about with the refined suggestions from Mask RCNN, but having multiple people annotate the same area. We had a bootstrap control where we don't apply Mask RCNN refinement at all. We just look at the classical image processing just to see if the refinement improves things. And then we have the unbiased control, where we say, just please annotate everything you see. You're not going to see anything to bias your perception of what the nuclei are. And again, now we have 2 controls, right? We have a way to measure the variability in the actual workflow we used. We have something to compare against if we ask people to, if we don't do the refinement, which again is something that we hypothesize would be useful. And then if we don't ask anything, we don't show the participants anything. I'll just fly by this area because it's fairly standard. Is we did truth inference. In this area, what I was showing is how do we compare actually how good people are doing, is we infer the aggregate label from crowds of people, right? So how do we infer that? We had to deal with a little technical issue, just related to the fact that first of all, we had mixtures. So we focus only on the bounding boxes. We ignore the boundaries for this truth inference exercise. So yeah, and we had to deal with various things related to positions and overlap of the bounding boxes. When do we consider these to be talking about the same class, and so on. And also, we had to impose a sort of constraint on basically, typically if the if 2 annotations are close together, if you do hierarchical clustering and you base it on the distance, if two things are close together, you would say, okay, these are probably talking about the same nucleus, right? So this would be the most straightforward way to do it. But the problem is that one person could be talking about two nuclei very close to each other. So if you look at this here, you know, you could have multiple people annotating the same field, and then you superimpose everything and try to cluster so that you say, okay, the truth is that this is a fibroblast right here, and this is another fibroblast, and this is cancer, and so on, right? But if you do the clustering using this raw data, you don't take into account the fact that someone might say, the same person might say that I think this is actually two fibroblasts from the same person. So we have to impose a constraint. It's a simple process, really, to make sure that the clustering process ignores two annotations if they're from the same person, regardless of their proximity in the spatial domain. And then once we have that, we basically match the anchors, which are the clustered potential locations of nuclei, and locations, I mean like bounding boxes, and participant labels, right? And by the way, knowing the location of the bounding boxes is a matter of taking the medioid of the different boxes that were clustered together. Anyways, to make a long story short, we once we have this matrix, we can just apply a simple expectation maximization type procedure to get a reliability matrix of which participant is reliable for what classification, and we can use that for various things down the line. Okay, so now that we have the process, let's see if it works, right? The actual results. So the first thing we saw is detection accuracy. Are they detecting whether nuclei are present or not? Now this is a classification agnostic. This is just if there is a nucleus or not, and just we do this, you know, simple precision recall type of curve. What you're seeing here, the dots here, the NP is like the non-pathologist, JPs are junior pathologists, and SPs are senior pathologists. For all intents and purposes, junior and senior pathologists are clumped together for downstream applications, but we also show them separate for you to see that there is a little bit of a difference actually between them. That junior pathologists tend to be more, they take more time in terms of the detection. Maybe not the classification, but maybe they're more free to spend time to detect. Again, this is all done on the multi-rater data set where we ask everyone, including the pathologists, to annotate the nuclei from scratch just to validate the approach itself, right? So this is the average precision. It's pretty good. And then you can see that there is a little bit of clustering within the pathologist by level of expertise. We also look at the classification accuracy. And what you're seeing here is that when you look at the micro average and macro average, you get different stories. If you do the macro average, which is basically gives equal weight to all of the potential classes, including the difficult ones, including the ones that are uncommon, you see a noticeable drop in the accuracy. But if you clump things together into super classes, like everything cancers together, everything tumor infiltrating lymphocyte together, everything stromal together, you see a much better story in terms of the accuracy, and also, you know, both and if you focus on the common things, so if you look at the micro versus macro average, you get a better accuracy, and if you use super classes compared to classes, you also get better accuracy. And there is no clear clustering by participant level here. So when I talk about accuracy here, essentially what you're seeing, this line here, this pink line here, is the truth, the crowd labels, the inferred labels from the non-pathologists compared to the truth inferred from the pathologists, right? So this is what this curve, this is how we are able to get sensitivity specificity type of curve, right? And then we can, when you actually cluster the participants, you do a multi-dimensional scaling type of linear clustering of the participant labels at the level of superclasses. So most of what I'll be talking about down the line is based on 3 superclasses: tumor, stromal, and TILs. We ask people to annotate whatever they see, but later on we sort of just clump the things that are relevant together. First of all, you can see that there is some weak clustering. by that pathologists and non-pathologists really have some differing perception of the nuclei, not strong, but it is there. And then we ask the question, well, if there is a little bit of difference between the two, how many non-pathologists do we need if they are not going to be supervised? Now, bear in mind, the main project is we actually look at the non-pathologist predictions, and then we have a pathologist go back and make sure that they're correct. But we ask, if we want to take this to a next level or a next project, and we want this to be completely agnostic process where there's no pathologist involved or they're minimally involved, how many people per high power field do we need to annotate the same area, right? So the same high power field, how many non-pathologists do need to annotate it for the wisdom of the crowd to be good enough? And then what we find is that this is quite dependent on the class, and that fibroblasts, for example, you need up to six or more non-pathologists to annotate the same area for the combined truth to be reliable. And then we also measure the effect of algorithmic suggestions that we show the participants. Now, this is the question that I'm sure is on a lot of people's mind. Are we actually biasing people? So here I'm showing one example. This is just a cherry-picked case. It's not like what everything is going to look like, but just to show you what the same person sees and annotates as an example. So here is from one participant where this is the unbiased control. This is the bootstrap control. The dots here are boundaries that we obtained by them just clicking, as opposed to drawing, because they don't draw anything. Everything else is drawn bounding boxes. And then when you look at the evaluation data set, which is after you smooth out the machine learning, the image processing with mask RCNN, you can see that the boundaries are especially improved. They click on a lot more boundaries as being accurate from what they see. And then we can also see how well the smoothing, the nuclear segmentation smoothing with mask RCNN does. We can see that the acceptance rate, whether among the non-pathologists or among the pathologists, increases when you do the smoothing with mask RCNN as opposed to just the heuristic image processing. So there's a significantly increased acceptance of the boundaries that they click and they say, yes, this is accurate. And the ones they do click are also more accurate. And the way we validate this is we have a limited data set where we ask someone to do everything manually, every segmentation boundary manually as a pathologist, so that we can actually show here that even when even the ones they do click, they're actually on average more they're more accurate than if the ones they do click are more accurate than the ones they don't click. So they do have a selection pressure, essentially. It's almost like, think of evolution or survival of the fittest. So you have a lot of boundaries, let's say 60% are inaccurate, 40% are accurate. Here we show that the 40% they do click are accurate, right? And we show that we ask the question, are they biased in terms of the classification? So far we've shown the segmentation boundaries. Are they biased in terms of the classification? And then what we show is that the pathologists, they're pretty, they don't change their classifications almost at all, right? So whether or not you show them the bootstrap control or the evaluation data, so whether or not you show them boundaries that have been smoothed, like the classifications that have been improved by the mask RCNN smoothing, or the original classifications that are like typically worse, they don't change their mind. They know what the right class is. They're pretty stable. Non-pathologists, they actually, we show that they get biased, right? But they get biased in a positive way. Maybe the difference is not very clear here, but if you look, for example, at the between, hold on a second. So here, this is looking at Cohen's kappa, which is like the variability between the non-pathologists. And then you can see that the variability among them decreases when we show them the improved classifications with mask RCNN smoothing, and their classification, the accuracy of their classification. If you look at the ROC curve right here, their classification itself improves. Maybe it's clear here for the macro average as well. So here is the unbiased, where they don't see any computer telling them where what the potential classes are, versus when we tell them we think this is a lymphocyte. What do you think? This actually improves their accuracy, and accuracy here is compared to the pathologist once again. So I think I'm going to go ahead and stop here. I don't want to overwhelm people. I want to leave time for discussion. So I won't get to the time to discuss the other paper, but I would like to hear what people think and maybe answer any questions. And if not, maybe I can go on.
Brilliant. Thank you for a great talk so far. We've got a couple of questions in the chat. So Mustafa has asked, did you use different data sets for control tests or considered off time between the annotations? It seems that you used the same data, but what was the order of control tests? I don't know if you want to speak a bit more about that, Mustafa.
Yeah.
Right.
Yeah. Hi, Mohamed. Great talk. So just wonder what was the off time? How much time did you consider between the different control tests, and what was the ordering of that control list? Did you do the unbiased control first and then go to the AI assisted ones, or the other way around?
No, this is an excellent question. So no, we started with the unbiased. We made sure that this had to be the case. No participant was allowed to, or at least they, you know, as long as they follow the instructions, but we made sure they did, is that they did the unbiased first. Now, in terms of the duration of the washout, we didn't enforce that very strongly, but on average, it was at least a week, just the time it takes, because they would annotate fields, and then we tell them, okay, just hold on. So the way this worked is we would ask them first to annotate the different fields from the computer-assisted ones to learn how to do the process. So there's a training process at first, just what the classes are and so on. And then we tell them, okay, while we give you feedback, please go ahead and annotate the unbiased one. So they would work on this one. And then we'd say, okay, now let's have a session and we discuss you know, your performance on this. So the unbiased was like before, there was an enforced gap a little bit between the two, but how long it was not enforced. So that you may argue that they may remember the field, but they wouldn't remember any suggestions because they did the unbiased first. That's maybe the main thing. The main disadvantage of what we did, I guess, is if they remember something from the bootstrap field of view and they move on to the evaluation data set. But this is quite unlikely because they finish the entire set of fields before they move on to a different control experiments. So it's not like they would take the same field and they say, okay, now it's unbiased, now it's slightly biased, now it's strongly biased. It would be like, let's do your unbiased controls first, and then let's, you know, talk about what you think, and if you have feedback, blah, 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 and then do all of your bootstrap controls, all of the different fields from different slides, and then you go back to do all of the evaluation data set. And to finish all of the fields, it takes a minimum of 1 week. On and off, if you do like an hour a day or so. So that's kind of what I would give here.
Thank you.
Brilliant. Thank you very much. So we've got a few comments from Nasir in the chat. So Nasir said, "Great talk. Thank you very much." He apologised that he's got to leave now, but he wanted to ask, what are your thoughts on opening up crowdsourcing to non-medical folk?
I'm ambivalent to it. Sorry, I had a stroke there. I don't think there's any strong objection to involving non-medical folk. The main issue is with the medical students, there's a lot of things that we take for granted that they know, but they only know because they've had you know, a year or two of training. So we don't have to explain what is a cancer nucleus, what is normal, what is fibroblast. We just have to tell them just the annotation process and the technical side of things in terms of the software. So with non-medical folk, it will be they will have to have that introduction, and of course with non-medical folk as well, especially if you don't validate, you get to the issue of you know, retention and things of that nature, just given that it's people may not have as much interest in that type of thing. So you know, this also opens the question of incentives. So for our projects, the incentives were non-financial. it was mostly that they would be authors on the data set that eventually gets published. And if they make enough contributions, they would also be authors on the paper. This wouldn't necessarily be a strong enough incentives for non-medical folk.
Makes perfect sense. I suppose another question would be, so how do you expect now with the presence of more deep learning algorithms to actually be able to perform? So obviously you could use, for instance, NuCLI if you had clicks, but also stuff like HoverNet where you could potentially get better priors, I suppose, for the annotation process.
Yeah, no, absolutely. If I were to do a second project where I want to annotate a different data set, the initialization process here, the main purpose of this is to generate data for sure, but there's a big chunk of what we're doing is it's a validation study really of the process. So using something that is a little bit on the less accurate or on the more difficult end is part of the process itself. It's part of what we're trying to show. In future projects, I would say, we should absolutely use something like NuCLI and even improve the segmentations that we initialize with. Use deep learning based segmentation methods or transfer learning from other cancers. Absolutely, I think that would be a perfect way to extend this if we want to generate other data sets. The one way that I'm not sure it would be very easy to extend is the classifications, because we inherited the classifications from low regions. That I think is something that I haven't seen much used in the literature, and I think would be pretty useful in getting people a starting point.
Do you have any more questions from the from people on the?
Yeah, I have a question.
We've got a question from Mr. again.
Yeah, thank you again, Mohamed. So was this not
Sure.
more of a general comment, or have you ever seen any kind of research like this? So I was wondering, how is the performance of crowdsourcing labeling? I'm not talking about dataset curating. I'm talking about labeling data set by crowdsource, not necessarily medical staff or medical student, but not pathologists, and compare that with senior pathologist performance on external data set. I mean, not using the aggregate knowledge of the same senior pathologist as the ground truth. You know what I mean? So taking the ground truth from somewhere else and see how those pathologists perform against the crowd, what do you call it? The collaborative annotation of large number of people.
Yeah, it's a fair question. I don't have very strong answers to that, and I would say that it's highly context dependent. So it depends how much data you need, really. If you need a small amount of data, and you're willing to train efficient models that take less data, I would say go ahead and use pathologist data directly. And because it takes some effort, unless someone comes up with a way to make this a very seamless, like crowdsourcing based annotation, a seamless process where think of a random, oh, I want to work on placentas. All right, you initialize, click, click, click. Now you got like workers who are, or medical students who are waiting in some sort of queue, and then they do the annotation. Unless you have that streamlined kind of process, and you don't need that much data, then you should probably consider having pathologists. Now, I was involved in a project where the crowd, quote unquote, was the entire pathology department in a place I was involved with. Now I can't say a lot of more details because it's not published, but basically, the entire crowd was like all of the residents somewhere, and you have a crowd of like 30 people that you didn't need to recruit, you didn't need anything. They can actually sit, together in the same room, and all of them just get you all the data you need. But then, of course, in that case, they're all residents, so if you're looking for something highly specialized, then maybe you can argue that that's still a form of non-experienced or less experienced participant crowdsourcing, and then you can make use of all of the techniques that we talked about here. So it's the applications here aren't necessarily limited to using what is that Amazon type of thing where you can get workers to I forget the names, it escapes my mind. But it's not necessarily limited to that. It's anywhere where you can have a bunch of people, pathologists or juniors or whatever, in a place and ask them to generate a lot of data in a short span of time.
Brilliant.
I hope that answers your question.
Yeah, thank you.
Okay.
And can you just remind us again how many nuclei are actually annotated as a result of this project?
So it depends on what you define the number, but we have 50. How many? I think so. It's 200,000 total with the multiple rater and so on. But then it's divided into different. If you want the single rater that are corrected, like the highest level of accuracy, I think these were on 70,000 or something on that side, but I forget the exact number. Oh, by the way, I need to plug in the website here. Is please everybody, if or anyone who's seeing the recording, you can go to this website. This is where the data set is published, and you can see the data generation process, the format, and then speaking of which, the single rater, we have about 60,000 nuclei, just to answer your question exactly. That's the one big data set, but the total is about 200,000 when you include everything with the control experiments and everything.
That's fantastic. I can imagine that's going to be a very, very useful source, especially in our lab. So, yeah, I mean, has anyone else got any other questions on the internet? If not, I suppose we can come to a close. But that was a really interesting talk. Thank you very much.
Sorry.
Oh, we've got one final question from
I just wondering,
yeah, sure.
What is the licensing on this data set? Is this like a permissive data set? I haven't come across any licensing information on the website.
Did you catch that question? It was about licensing.
Yes,
yeah,
it's about licensing. So this is 100% unrestricted license for every single application. I think it's called a CC0. Where is it? Let me just. Yeah. Okay. So it's fully unrestricted. Use it for anything you want.
Okay, that's cool. Thank you.
Brilliant. Yeah. Well, yeah, thank you very much. Yeah, it was a great talk, and I hope everyone else has enjoyed it. And yeah, brilliant. And a reminder that we have our next talk at the same time, but on a slightly different day in 2 weeks' time, Tuesday, the 6th of December. And we've got James Dio from Harvard Med School doing a talk. But that's brilliant. Thank you very much, Mohamed.
Thank you very much, everyone. It was a really nice meeting you, and thank you for the kind invitation once again.
Not at all.
Thank you. All right. Bye, everyone.
The rest of the day.
Thank you. Bye-bye. Thank you.
Was he about to talk?